Hello everyone, welcome to Pathomad. Today let us study about malignant melanoma. Malignant melanoma is a skin cancer that arises from the melanocytes. Melanocytes are present in the stratum basal layer. So these are the layers of the epidermis. Firstly is the stratum corneum, below it is the granular layer, next is the spinosum layer, whole length is the spinosum, this is the basal layer. In the basal layer, this is the melanocyte. So this malignant melanoma, this particular tumor, it arises from this melanocyte which is present in the basal layer of the epidermis. So malignant melanoma is the third most common skin cancer after squamous cell carcinoma and basal cell carcinoma. 10 to 15% of these affected individuals they pass on these risk as an autosomal dominant trait to their offsprings. There's an entity called familial dysplastic nevus syndrome, which is an autosomal dominant inheritance. In this particular entity, there's presence of more than 50% of dysplastic nevus, and these patients also co-inherit malignant melanoma. Dysplastic nevus is considered as the most important precursor for this malignant melanoma. The rest majority, like 80 to 85% of the cases, the vast majority are sporadic. Sporadic means does not pass on does not run in the families. They are acquired mutations because of exposure to the UV radiation. This is strongly linked to exposure to UV radiation, the acquired mutations, especially UVB radiation, which has a nano wavelength of 280 to 320 nanometers. They induce damage to DNA. This particular radiation oncogenesis I have discussed in my previous video of chemical and radiation oncogenesis. For in detail understanding, you can refer to that video. So the UVB radiation, it induces damages to the DNA and form thymine dimers. The pathogenesis is by forming thymine dimers. For example, this is the double-stranded DNA. It unwinds the double strand, and between the adjacent thymine residues, they form a dimer. So it prevents binding with the opposite DNA strand, and hence it induces damage to the DNA. The accumulation of these mutations in the melanocytes over time, if at all the DNA is damaged and it goes unchecked and the cell proliferates over time, it ultimately leads to cancer. And fair skin individuals are at higher risk for this radiation induced cancer because the fair skin individuals, the melanin content is low. Because of low melanin content, the protective action is lost, so there's higher damage to the melanocytes by this radiation. Let us study about the molecular pathogenesis, by what pathways this malignant melanoma has occurred. It is mainly driver mutations. Driver mutations means the occurrence of mutation itself starts the pathogenesis. That is meant by driver. It starts the car. So the occurrence of mutation itself, it starts off the cancer. There are three pathways. First one is the mutations that affect cell cycle control genes. Other are the activate the pro-growth signaling pathways, and the other is activate telomerase. So that affects cell cycle control genes means it affects the tumor suppressor genes. So the checkpoint, the break on the cell cycle is lost. Other is the activate the signaling pathways, higher activation of signaling. Other is the activate telomerase. So the cell becomes immortal. Let us discuss each one of them in detail. Firstly, let us see the mutations that affect cell cycle control genes. Here, there's a gene called CDKN2A gene. CDKN2A gene is the main gene that gets mutated. CDKN2A gene gets mutated. Because of this gene mutation, there's loss of P16, P15, and P14 ARF mutation. P16 and P15 you must have studied in the earlier chapter, first chapter of cell cycle. Here, this is the cell cycle. This is the image of the cell cycle. From G1 to S phase, it is mediated by cyclin-dependent kinase 4 and 6. See, binding of cyclins D. The cyclin D binds to CDK4 and CDK6, and this is how the cell cycle moves from G1 to S phase. Here, the RB protein gets phosphorylated. Phosphorylation of RB protein, it moves the cell cycle from G1 to S phase. Now this cyclin B and CDK4 and 6, these are inhibited. CDK4 and 6, these are inhibited by P16 and P15. 15 and 16 play a role in they put a brake action on the CDK4 and 6. When this CDK4 and 6 put a brake on CDK4 and 6, the cell cycle stops here. It stops here. And also the RB protein is not phosphorylated. It holds the cell cycle in this G1S phase. It holds the cell in G1S phase. When this P16 and P15 are lost, the brake the brake on the cyclins and cyclin dependent kinases is lost, and so the damaged cell, it does not get checked. It is not checked, and it is easily passed from the G1S phase. And the RB protein is phosphorylated without getting checked. It phosphorylates itself, and the damaged cell is passed through this checkpoint, through this toll gate. And this is how the damaged cell, it proliferates, survives and proliferates. So the P16 and P15, normally the normal function is to inhibit CDK4 and 6. It allows the RB gene to block, to check the G1S phase. When this P16, P15 are lost, they do not inhibit the cyclins CDK4 and 6, and the RB protein is not checked. It phosphorylates and moves the cell cycle from G1 to S phase. That's how the cell cycle proliferation occurs. The cell survives, damaged cell survives and proliferates. And the P14, also called the ARF, it normally inhibits MDM2. The normal function of MDM2 is to degrade P53, the guardian of the genome. So P14 ARF, when they are lost, the inhibition of on MDM2 is lost, and so the net effect will be increase in P53, because this is a mutation. This is a mutation. So mutated P53 is produced. The net effect of all these mutations are uncontrolled cell proliferation. This is the net effect. The other pathway, second pathway, is mutations that activate pro-growth signaling pathways. Pro-growth signaling pathways are, for example, this is the receptor on the cell membrane, growth factor receptor, which activate the RAS pathway, RAS PI3K AKT pathway. There's activation of this RAS pathway, RAS PI3 AKT mTOR pathway, they get activated. And also they, because of BRAF mutations, there's a proliferation. See, there will be aberrant increase in the RAS and PI3 AKT signaling pathways. Also, because of BRAF mutations, there's loss of PTEN's tumor suppressor gene. There's loss of tumor suppressor gene, which also contribute to this enhancement of RAS and PI3K AKT signaling pathway. When all the signaling pathways are activated, there's abnormal increased stimulation for the cell to proliferate more and more without getting checked, and hence it leads to abnormal cell growth, ultimately cancer. The third pathway is the mutations that activate telomerase. Here there is a concept. This is the structure of a chromosome. The ends of this chromosome, the ends of arms, each arm of the chromosome, they are called telomere. With as the cell ages, as it becomes old, the telomere, it decreases in length. It becomes short and short and short. Ultimately, it ends up. This is how the cell dies. So normally the telomerase, which is an enzyme, telomerase is an enzyme that adds nucleotides. It acts certain nucleotides, guanine, thymine, like that, cytosine, guanine. They add on this to the ends of chromosomes, and hence they stop the cellular senescence. It stops the cells being old or cells being quiet, and hence they ultimately lead to cell immortalization. Immortalization. Normally, the ends of chromosome, they are called telomeres. As we age, the telomere decreases in size and ultimately it ends. Telomerase, which is an enzyme. Telomerase is an enzyme. They add nucleotides to these ends of the chromosome, and hence it prevents the cells from becoming old or from cells from dying. So it stops the cellular senescence, and it makes the cell immortal. In our body, there's a gene called TERT gene, TERT gene. This TERT gene encodes for a catalytic subunit of telomerase. Catalytic subunit of telomerase. This is how the TERT gene is helpful to degrade. It degrades the telomerase. So the telomerase is not present in excess. So it helps the old cells to die. What if the TERT gene mutation occurs? When TERT gene mutation happens, the telomerase is not degraded. There's excess of telomerase. Excess of telomerase means it adds the new nucleotides. It keeps on adding the nucleotides to the ends of chromosome. In this way, the cells become immortal. In this way, cancer develops. This is one mechanism. This is the third mechanism. Coming to the clinical features, we are well versed with this ABCDE rule. A means asymmetry. For example, you cut or divide the tumor into two halves. Each half is asymmetrical. This half is different from this half. And irregular borders. The border is obviously irregular. Irregular borders. This color where you see for color variation. There is color variation. For example, here it is very dark, almost black or dark brown. Here it is light brown, almost pinkish. This color variation and the diameter being more than 6 mm and elevation or evolution of it increases in size or there can be an elevation of this lesion. Coming to the clinical variants, we have four variants, superficial spreading being the most common, lentigo maligna melanoma, acral lentigo maligna, and nodular melanoma. These show different growth patterns. Firstly, let us see about the growth patterns. In malignant melanoma, when you observe under the microscope, there are two growth patterns within the epidermis and dermis. Firstly, radial growth pattern. Radial means it grows laterally. For example, this is the lesion present in the epidermis. These are the layers of the epidermis: corneum, granulosum, spinosum, basal. It is limited to the epidermis. It grows even if it wants to grow, the lesion grows laterally within the epidermis only. This is a radial growth or the lateral growth. Radial growth. Other one is the vertical growth. Vertical means the name itself suggests it goes deep into the dermis. The lesion starts within the epidermis and it grows vertically into the dermis. This is the vertical growth. So these clinical variants, they show different growth patterns. The superficial spreading, the name itself suggests superficially it is spreading means within the epidermis. So it shows a radial growth. Other is nodular. Nodule means a big mass like thing like this. So it has to grow within deeper into the dermis. It shows a vertical growth. Superficial spreading, superficial, radial. Nodular, it should be deep, so vertical growth. The remaining two variants, the lentigo maligna melanoma and the acral lentigo maligna, it shows both growth patterns, radial as well as vertical growth patterns. Coming to the microscopic features of this malignant melanoma, most of the tumor cells they show nested pattern at the dermo-epidermal junction. And this is one such notorious tumor. It shows great microscopic variability. The cells they are larger than normal melanocytes, and the nucleus within the cell it shows like routine features of pleomorphism like hyperchromatism, irregular nuclear membrane, and the chromatin is clumped to the periphery of nuclear membrane. Here, one eosinophilic nucleoli is the characteristic among all. It can be seen or cannot be appreciated in some tumors, eosinophilic nucleoli, and some of the nucleus can show binucleation. This is sometimes present, and presence of mitotic figures. Presence of mitotic figures, it shows in any cancer, in any tumor, it shows the aggressive nature of the tumor. Presence of mitotic figures, and giant cells can be seen or may not be seen. Cells can be, and because it is a tumor of great variability, microscopic variability, the cells can be either epithelioid, means slipper shaped, epithelioid or spindle shaped with tapered ends. Let us see the microscopic features of each other variant. Firstly, the most common variant being the superficial spreading. Here you can see this is the layers of epidermis. This is the corneum, granulosum, spinosum, basal. This is the basal. It is confined. This is the tumor cells. These are confined within the epidermis only, does not grow beyond the epidermis. And the tumor cells, they are arranged like a nested pattern within the layers of epidermis, nested pattern. So this is given famously a name called buckshot scatter appearance. Buckshot scatter. I'll explain you what is a buckshot scatter. So buckshots are the pellets. They are pellets used in firearms. So this is the buckshot. This is the birdshot. Birdshots are relatively smaller. This is the buckshot. These are the pellets present within this plastic tube, which are used in firearms, in mostly used in shotguns. You fire it. These are the buckshots. So similarly, these are arranged in the form of buckshots. So hence the name buckshot scatter appearance. Buckshot scatter appearance is a name given to superficial spreading melanoma. It shows only the radial growth. Only the radial growth is seen. It does not move into the dermis, only it is spreading within the epidermis. And this is called nested pattern of tumor cells within the epidermis given a buckshot scatter appearance. Next variety is the nodular melanoma. As the name suggests, you can see here large nodule. No, it is present both within the epidermis and extending into the dermis. A huge nodule. This is a nodular melanoma. Other variety is the acral lentiginous. It has to show you both the radial growth and the vertical growth. It is spreading radially within the epidermis and also into the dermis. It shows both the radial and the vertical growth. And here and there you can see the melanin pigment. Within the acral lentiginous, there is another subvariety called mucosal lentiginous melanoma, mostly confined within the oral cavity. Mucous membranes like oral cavity, conjunctiva, they also show this mucosal lentiginous melanoma. Other variety is lentigo maligna melanoma. This variant is most commonly associated with dense lymphocytic infiltration. So this shows you both this superficial spread, that is the radial, and the deep vertical spread. Tumor cells are present in both ways, and here uh, this is a dense lymphocytic infiltrate, which is responsible. This lymphocytic infiltrate responsible for the tumor regression. Coming to the IHC markers, S100 being the most sensitive, it is the sensitive, whereas HMB45 is the most specific marker is the HMB45. There are other markers like Melanase or the MART1, CAM 5.2, KR67, Thuracinase. All these are the IHC markers out of which HMB45 is the most specific. This is the IHC marker. This brown deposition, brown one is showing positivity for uh, IHC HMB45. This is all you need to know about crisply about malignant melanoma. Thank you all.